phrasing. Okay, so here's the stuff I lectured about, and I really am, I'm not trying to be mean by skipping over it mostly and recording it. I really am concerned about having enough class time so that's all the stuff that you can hear me talk about, drone on about in the recorded lecture on D2L. And it's really pretty cool. Obviously, I like the topics. So this was as far as I got, and so this is where I'm going to begin. I have an apology to make. I posted the wrong notes originally for this chapter, and so I posted a new set today and sent out the notice, and I'm sorry about that. But some of you may be missing some of these notes there. It is available on D2L now, and so, yeah, sorry about that. So this is a good time to review how anterior pituitary hormones are controlled, and we're going to take as an example thyroid hormone, because we're going to talk about thyroid hormones, which is absolutely crucial for brain development in all the vertebrates, right? Without thyroid hormone, the brain is kaput and will not function. And if there's absolutely no thyroid hormone, the organism is doomed, not viable. So it's a crucial hormone for brain development, and so we'll talk about that some. Also, just to give you a little bit of history, the thyroid hormone was also the first hormone that was actually recognized as such. And that's because there's something cool about thyroid hormone, which is endocrine glands store, the thyroid glands store a lot of thyroid hormone at any one time, right? They don't want to run out, so they keep an ongoing supply. And in the 19th century, that turned out to be crucial to find that taking up thyroid glands from sheep, say, and grinding them up could be used to help people, to treat people, etc. People have tried similar things with testes, as a source of testosterone. And there's a pretty hilarious French physiologist in the late 1800s, who talks about taking dog's testes and mulching it up and then taking a liquid emulsion of it and injecting himself with it. And he was 72 years old, but now it was a wonder, right? Now he could go upstairs without catching his breath. He could work all night. It even cleared up his constipation, right? All this in the Lancet, he's talking about. And it was so absurd, so over the top, that people dismissed it, right? And he was his last name was Brown-Séquard. He was a very prominent physiologist, a great scientist. It's sort of a sad end to his career. And there couldn't have been any testosterone in there, because testosterone won't dissolve in water, for one thing. And the other is testes don't store much testosterone. As soon as they make it, it goes into circulation, right? So Brown-Séquard almost doomed the idea of hormones before he even had a chance to get started. So it wouldn't be until later when people started looking at thyroid. All right, blah, blah, I'm gone on too long. So, as always, the brain is totally in control of hormone secretion. Every hormone, any hormone, and in fact, any physiological process, the brain is in charge of it. And that makes plenty of sense, because the brain's job in natural selection is to gather information and decide when is the right time to release hormone and when is the wrong time to release hormone. And it doesn't matter what the hormone is, that's the job. So you can think of the brain as being a funnel for all the information about the world as best it can discern it. And as in the anterior pituitary, it'll be the hypothalamus that has neurons, neuroendocrine cells, that send their axons to the median eminence and release into the hypothalamic pituitary portal system, those capillaries that I talked about in the recorded lecture. Let's go back to them. Well, the neuroendocrine cells in the hypothalamus send their axons to the start of the hypothalamic pituitary portal system. They're neuroendocrine cells, so they're just like neurons. They have synapses and dendrites and all that stuff, action potentials, but they don't release their product into a synapse. They release it into the portal system, where it travels down to the anterior pituitary, where it runs into various kinds of anterior pituitary cells, some of which will respond to a given releasing hormone to release a tropic hormone, not tropic, tropic, and into general circulation. And while you cannot, the total amount of releasing hormone in general circulation that you can take from blood in the arm, it's too small to be detected, even by modern day assays. But the tropic hormones are the ones that we typically measure. So for thyroids, the thyroid gland in particular, it is neuroendocrine cells in the hypothalamus produce a product called thyrotropin releasing hormone, or TRH. And it's called a releasing hormone, if this helps you any, because it's going to prompt the anterior pituitary to release something. In response to that, certain anterior pituitary cells are geared to respond to the TRH by releasing a protein hormone. These are both protein hormones, thyroid stimulating hormone, and at least that name makes sense. Its job is to stimulate the thyroid. That's the tropic hormone involved. And the thyroid gland, in turn, releases thyroid hormones that affect target cells. Several things about this. Number one, the thyroid gland, really there are two principal hormones that we lump together as thyroid hormones. And I guess I want you to know what they're called, but we're never going to ask you, which, let's see. And they're called, what they have in common is they all require at least three atoms of iodine in their structure. So one's called triiodothyronine, or abbreviated T3, and the other's called tetraiodothyronine, T4, and for a while it was also called thyroxine. And T4 is the principal hormone, but T3 does the job too. Okay. And we'll always lump them together. The levels of thyroid hormone are regulated, as all anterior pituitary hormone secretions are regulated, by negative feedback. That is, when the thyroid glands release their thyroid hormones, it feeds back and informs the hypothalamus. Thyroid hormone has been released. You can cut off the flow of TRH. And it also informs the anterior pituitary. There's thyroid hormone out there, so now you can slow down the release of TSH. And it's this negative feedback that keeps thyroid hormone levels from getting too high or getting too low. If they get high, negative feedback turns off the flow. If it gets low, negative feedback is released, and now the system starts calling for it again. And it's a peculiarity of the thyroid glands that measuring thyroid hormone itself is not necessarily a good indicator of anything's wrong. Because if the thyroid glands are having a hard time making thyroid hormone because of negative feedback, there's just going to be more TSH release driving the thyroid glands. Everyone get that? And so the better indication of how well your thyroid gland is doing is to measure levels of TSH. Because if the levels of TSH are high, that indicates the thyroid hormone is being sluggish, and there might be a problem in your future, right? And so that's the diagnostic to look at. Why don't we measure TRH to see how your thyroid system's doing? Justin?
Can't detect it.
Pardon? You can't detect it. It's released there in the hypothalamic pituitary portal system. By the time it enters the entire bloodstream, there's too few molecules to detect. Okay. And as we're going to see in a moment, if there's some problem that the thyroid gland absolutely cannot produce thyroid hormone, then levels of TSH will go through the roof. And TSH has several effects, but mainly it's pushing the thyroid gland to grow in order to make more thyroid hormone. But if the gland can't make the thyroid hormone, then TSH levels stay high and the thyroid grows bigger and bigger and bigger, and that can be a problem, about which more in a moment. And the other thing that I need you to know about thyroid hormones if we look back at their structure, so this is the structure of steroid hormones. They all have this 4-carbon ring backbone derived from cholesterol, typically from the diet. And the steroid receptors are very closely related to the thyroid hormone receptors, right? And so probably thyroid hormone receptors, probably the first steroid receptors were retinoic acid that arose in evolution. And the second steroid receptor that arose was either the steroid hormones of thyroid hormone, thyroid receptors. Anyway, and so you can see that the thyroid hormone is not structurally that close to steroids, but sufficiently close that it still binds to the same kinds of receptors. But there's one difference between the thyroid hormones and the steroid hormones. Whereas steroid hormones can pass through membranes very readily, the structure of thyroid hormone is sufficiently different that it can't. So target cells can only respond to thyroid hormone if they have a transporter that takes extracellular thyroid hormone and inserts it inside the cell, where it can then interact with the thyroid receptor. And then, as with steroid hormones, the thyroid receptor complex now binds to DNA and affects gene expression. How's that? Now, the good news is, because thyroid hormone has such widespread effects, the good news is that almost every cell in the body has thyroid hormone transporters to respond to it. The bad news is that that's one more link where thyroid hormone signaling can go wrong. So there's several places where thyroid hormone signaling can go wrong. Number one, right, there might be a problem making the thyroid hormone itself. Right? Number two, there might be problems with the thyroid hormone receptor, the cytoplasmic receptor that is needed for it to have its effect on gene expression. Number three, there might be something that go wrong with, or you might have a gene that gives you an ineffective thyroid hormone transporter, and any of those things will interfere with thyroid hormone signaling. You get that? And if they happen early in development, the brain is in trouble. If it happens in adulthood, things aren't quite so bad. But what happens in adulthood is, we'll get to that in a moment. Questions about that? If thyroid hormone signaling is messed up somewhere, there'll be higher and higher levels of TSH. The thyroid gland, which is situated in your throat, will get bigger and bigger, and pretty soon you'll be able to see it. And once it's visible outside, it's called a goiter, about which more later. How many of you have ever seen anyone who had a goiter? Oh, more than I thought. Okay. Okay. Well, they used to be quite common, but about which more later. So one interesting area where thyroid hormone is involved is in metamorphosis among vertebrates in the amphibians. So it turns out that thyroid hormone is crucial for amphibian development and crucial for the metamorphosis from the larval stage to the adult stage. So here we're looking at levels of various things. So the retinoic acid receptor is expressed very early on. Why is that? Why is it expressed very early in development? Yeah. It's a crucial cue for signaling posteriority, right? Because there's a higher level of retinoic acid in the posterior end than the anterior end. So, of course, the receptors have to be there too. And here we're keeping track of thyroid receptor alpha and thyroid receptor beta. And notice that the receptors are in place before any thyroid hormones have been produced. Just a peculiarity of it. So the receptors are sitting there waiting for that signal, and when the thyroid hormone arrives, they start pushing into metamorphosis. This means that if you want to push a frog into metamorphosis faster, all you have to do is put some thyroid hormone in the water, or inject the animal with thyroid hormone. And it already has the receptor to respond. It'll start transforming. And another thing that I think is super cool, there are some frogs where they come out, they don't go through a tadpole stage, right? There are some tree frogs, for example, where the animal comes out of the egg already, with legs in terrestrial form. How does that happen? In those species, mom puts a bunch of thyroid hormone in the egg when she lays it. And so they're undergoing that metamorphosis inside their egg and come out, cute little, tiny little frogs. So thyroid hormones, once they start getting secreted, they will push this transformation, and then they go down again, and they never go back to zero, but they stay very low. So one thing that's interesting about this is this means that in these animals undergoing metamorphosis, apparently there's no need for thyroid hormone early in development for brain development to proceed, right? The aquatic stage of the larva, they have brains, of course, but they're not as dependent on it as other terrestrial vertebrates, and certainly mammals. So here's a similar chart for humans, in this case, where we're keeping track of fetal development in weeks, right? 40-week prenatal development, and then years. So it's not to scale. And here we're keeping track of maternal thyroid hormone production when things are going well. And as that happens, you'll see more T3 appearing in the fetal brain, more T3 binding to nuclear receptors in the fetal brain, as well as in receptors in other organs. The thyroid gland is present in the fetus at this stage, but it's not producing any thyroid hormones yet. So the production of thyroid hormone from the fetus doesn't happen until about the halfway mark, about 20 weeks of age, 20 weeks of development. So until that point, the fetus is dependent on maternal thyroid hormone for proper brain development. If mom happens to be hypothyroid, so she's not making much thyroid hormone, brain development would be stunted. And you can take a look, and then by 20 weeks of age, the fetus can make its own thyroid hormones. And you can see here they're keeping track of the various processes that are known to be affected by loss of thyroid hormone at that point. So just about everything in development is affected. It's not that without thyroid hormone, none of that stuff happens, but it's greatly depressed. And so it sometimes happens that the fetus itself is not able to make thyroid hormone or is not able to respond to it. We talked about the various places where things can go wrong. That can happen, various recessive traits. But that is a terribly, and that will definitely need to be addressed. But as long as mom was able to make thyroid hormone, development proceeds. Where things really go wrong is when the mother is hypothyroid during pregnancy, nobody notices it, and the fetus is unable to make its own thyroid hormone. And in those cases, they're severely affected. I'll talk about what sort of effects in a moment. And it's very difficult to recover. In the case where it's only the fetus itself that can't make the thyroid hormone because of maternal thyroid hormone, those kids have very good prognosis because at birth, as long as they're identified, we now screen all babies for thyroid hormone. As long as they're identified, they can take thyroid hormone the rest of their lives and be fine. Yeah.
If a
mom has Hashimoto's disease, tell us about that. That's an autoimmune disease that...
It's later on. Yeah. Well, one thing is how the boys affecting it.
How does it affect pregnancy? Well, first of all, there's a higher rate of the pregnancy being lost, but I don't know a lot about Hashimoto's disease in particular. But if the thyroid hormone deficiency is sufficient, the child will be, greatly affected. I understand. I think Hashimoto's disease tends to start in adulthood, right? So, because like most autoimmune diseases, and so for the person themselves, a thyroid hormone supplement can basically make things go away, make the problem go away. And what else do I need to say there? I'll just remind you, congenital hypothyroidism, that just means, congenital, I'll remind you, just means present at birth. Doesn't mean genetic, maybe, maybe not, but it just means present at birth. And so congenital hypothyroidism is totally treatable. And here's just another look at levels in humans in terms of, here's where the fetus starts making its own thyroid hormone. And here you can keep track of serum levels of T3 and T4 and how much is bound in the cortex, how much in the choroid plexus, and total T receptor protein found, etc. If in adulthood or in development, if there's some problem with thyroid hormone signaling, then TSH levels get really high. The thyroid gland is being stimulated more and more by the TSH, but it can't produce the thyroid hormone, and therefore TSH levels stay high. Then you'll have a condition occur like this with a goiter, known as a goiter. Goiters used to be very common. It turns out that when iodine was discovered in the late 19th century, it wasn't long before people realized that there was a relationship between iodine and goiters. And it would be once they discovered the structure of the thyroid hormones that people would understand this. For the most part, goiters are caused by, until recently, until the 20th century, most cases of goiter were due to a lack of iodine in the diet. So back in a time when everything you ate was grown within a 3 or 4 or 20-mile radius of where you happen to live, if you happen to live somewhere where iodine was really very little of it in the soil, you weren't going to get much of it. And in that case, you would have the problem making thyroid hormone and all the things that would happen to it. When it was indigenous in places where both mom and the fetus was unable to make much thyroid hormone, it would cause a condition that was termed cretinism. And no one knows where that term cretin came from. It's thought to be that it may be, from the French Christian, right? They're just, a child of Christ and therefore worthy of concern, but no one knows. It's now considered a disparaging term, of course. And so you won't see that. People won't talk about cretinism, they'll talk about hypothyroidism. But in those parts of the world where there wasn't much iodine in the soil and therefore not much iodine in the diet, the proportion of children with this condition was quite high. And so you can see there in this photograph some of the conditions. Without thyroid hormone, the skin starts getting really thick and waxy and kind of swollen. Without thyroid hormones, growth is stunted, so they tended to have stunted stature. It caused a thickening of the cranium, of the face, etc. It was accompanied by a goiter. There was a time in polite society where if you had a goiter, you might wear a special collar around your neck so that people wouldn't see that you have a goiter, right? If you could afford a collar. And most importantly, for our purposes, it's accompanied by severe intellectual disability. So most of the children with this condition would never learn to speak, for example, and would basically need to be taken care of all of their lives. So this condition is relatively rare now, but it used to be quite common, including here in Michigan. So in the United States, there was once a goiter belt that went across the northern United States, where here we're keeping track of areas with really low iodine in the drinking water, which means in the soil, and areas where the goiter frequency was more than 5 per 1,000 cases. There were some areas where 20% of the children in the area had goiters. which was a pretty good indication that they'd been suffering from thyroid hormone insufficiency and therefore were intellectually disabled. So, and it's been estimated that worldwide, hypothyroidism is the single most important cause of intellectual disability in the world today. It's virtually abolished in Western nations because in the early 20th century, when people realized that you had to have iodine to avoid goiters and to avoid cretinism, then a campaign in the United States began putting iodine in the salt so that everyone would get enough. And so the Morton Salt Company of Detroit, where there are vast salt mines that are being harvested even today, they began introducing iodized salt. And the goiter belt disappeared. It drives me crazy to know that you can find people who, conspiracy theorists who don't want that adulterated salt. They don't want iodized salt to take home and, all you can do as a scientist is you show them the data. You can't make people believe it. Yeah. Did you have a question? Oh, I'm sorry. Okay. But back there, did you? Okay. So what was the other thing I want to say? Oh, yeah. And here, I thought it was pretty cool. There was a Michigan physician. So in World War I, a lot of things happened in World War I where for the first time, millions of people were being processed by a government because the draft came and they were going to ship all these young men off to fight in Europe. One of the things that arose was the need for intelligence tests, right? So IQ tests, they actually began in France in the late 19th century, but they got a big boost when the Defense Department needed to screen millions of men to see which of them were, possibly officer material and which were so unintelligent that they couldn't even send them off to fight. And another thing that arose was people keeping track of, look at this, that 30% of the registrants in the military from his area had goiters. And at that time, that disqualified them from service. And that got him, thinking about, well, why are there so many goiters here? Anyway, questions about that? Okay. Well, I'm a little bit perplexed. I wrote a Kahoot this morning about thyroid hormone, and I cannot find it on my Kahoot site. So I'll find it, or if I have to, I'll just write it again. But it will come. But so we'll do it next time. Questions about thyroid hormone, etc. It just has a pervasive effect. Yeah, just.
Well, the frogs that are given thyroid hormones, they usually receive it later if they're given it early element. Do they smaller? Is there some difference?
Yep. As you'd expect, they have less time gathering food and growing as larvae, as tadpoles, and so you can,
Chiefs don't like their own structure.
Yeah. No, the receptors are there. They'll respond to the signal. So it's the only reason they haven't turned into adults is because the thyroid hormone hasn't come around yet. Allison?
Is there anything bad about having too much thyroid hormone?
Well, there is, yes. Let's see. So to make all this a little more clinically relevant to adults, first of all, if your thyroid gland stops working as well, you're going to notice several things about it. One is that, you're going to feel chillier than you used to in various rooms. That's okay. Another is your thinking is going to slow down. And if it's severe enough, you might really become punch drunk, right? In terms of, because the thyroid hormone isn't doing its thing in the brain. And one way to indicate hypothyroidism might be, the knee-jerk reflex, tap the knee and make it, well, it's visibly slower in people who don't have enough thyroid hormone. So that was one reason. You might wonder, why did they do that test? Because if they do it right, it always jerks. Well, the question is, how quickly does it happen? Anyway, if you have hyperthyroid, that can also be a problem of course, because then, among other things, may become feverish, and I actually ought to remember the names of the syndromes or hyperthyroid, hypothyroid, but I don't. How do you know? Pardon? Graves' disease is hyperthyroid, and that too will interfere with thinking. Right? People become manic.
This anything in development that which is thyroidism?
Not that I know of. I don't know that it's detected. So does anyone know? Hyperthyroid fetal development? I mean, really brainy folks, not that I know of. Okay, well, I'll save up my lollipops. We're going to shift gears a little bit. We're still going to be talking about hormones. And in terms of your reading, so I just finished talking about the material that's in that Word file and under perusal that you could read. And now I'm going back to the original textbook in chapter 7, where we're going to talk about sexual differentiation. And again, those of you who've taken hormones of behavior, this will be somewhat repetitive for you. But hopefully, sometime second time around, you integrate a lot more. So sexual differentiation is the process by which, within a species, some individuals go down the pathway of developing as males, and some go down the pathway of developing as females. In vertebrates, this is totally dependent on gonadal steroid hormones, and we'll talk about that in some detail. Just to warn you, we'll also talk about sexual differentiation in insects, because it's very well understood in Drosophila, and just to let you know ahead of time, in insects, it has nothing to do with hormones, right? Sexual differentiation in insects is strictly due to cell autonomous response to sex chromosome. Okay? So be ready for that. And who first figured out this business about X and Y chromosomes and sex, right? Everyone knows in mammals, 2 X chromosomes will result in a female, XY chromosome will result in a male. Do you know who first figured that out? You don't know her name? Nettie Stevens. You don't remember Nettie Stevens? She never got a Nobel Prize, right? No, but she was the first one to figure out in insects about X and about long and short, that's what she called them, chromosomes, and the relationship between long and short chromosomes and sex in insects. Later, other people like T.H. Morgan saw that the same thing was happening in flies. Someone else named them X and Y chromosomes, and there it all went from there. But of course, Nettie took forever to get a faculty position. She was her parents. Her father was a carpenter, so she was from rural Vermont. She worked as a school teacher for years to save up enough money to go to college, to go to graduate school. And by the time she got a faculty position, I believe at Bryn Mawr, she was in her mid-30s, and she got breast cancer and died. So look up Nettie Stevens sometime for an interesting story. And was totally forgotten. Among things, just to let you know that sexism is, has always been with us, she sends her paper off describing her results to a guy who's a mentor and editor of a journal, and he just happens to have a paper with similar findings from a different insect himself that he's working on. And somehow her paper doesn't get published until something like 8 months after she submits it, and his paper gets published a month after he submits it, and it's actually published before hers. And he just happens to be one of the editors of the journal. Yeah. Anyway, at least he cited her paper in press, right? So there you go. Okay, so just as I said in the lecture, what makes hormones perfect for controlling metamorphosis in insects is the fact that the hormones go everywhere. So Have any of you noticed that, have any of you ever seen a clock, a town clock, that signaled the hour with flashing lights? No? How about releasing odors? No? No one ever made such a... How about... how come? Well, because those are lousy mediums to communicate with lots of people at once. Sound is the perfect medium for clock towers. The reason they use sound is because sound goes everywhere. It goes around corners. It can even go through walls, right, into buildings. So if you want to synchronize a big population in a particular area, sound is a perfect medium for that. Likewise, if you need to coordinate lots of different activities in the body, hormones are the perfect medium for that. Because as soon as the substance goes into circulation, it basically gets everywhere in the body in short amount of time, and therefore can cause very different things to happen in very different parts of the body. For insects in metamorphosis, that yin-yang of juvenile hormone and ecdysone means that as long as juvenile hormone levels are high, when the ecdysone signal comes, the animal will simply make a new larval skin. But once juvenile hormone levels fall, then the next stroke of ecdysone will be telling the body to start making a different body, the insect to make a different body, first the pupa and then the adult body. Likewise, in terms of sexual differentiation, so many different parts of the body are different in the two sexes that it makes sense that hormones would be the signal that would coordinate those changes across lots of different cells. And the other thing that will happen there is that many different cells in different parts of the body will be responding to the hormone, but they don't all do the same thing. Each of them takes a different pathway in response to that same signal. So, as I said, steroid hormones are going to be important for this. So a little bit of terminology, because I tend to go back and forth. Androgens represent a class of steroid hormones, right? So there are many of them. And we'll only talk about a couple, but there are a lot more than that. The most common, the most important androgen is testosterone, which is a principal secretion of the testes in mammals anyway. And in general, androgens are higher in males than in females, although females also have to make androgens, and they're important for them too. Estrogens represent a class of hormones that are like estradiol, and progestins are a class of hormone that are like progesterone. Estrogens, the name of estrogens has kind of a misogynistic, has a misogynistic base. It suggests that frenzy, because it's associated with females being in heat, showing, so-called frenzied behavior. Progesterone at least has a nicer meaning. Progesterone is progestational. In general, across the mammals anyway, progesterone promotes pregnancy, promotes gestation. And I need to get that out of the way because I'll tend to go back and forth about that. So sexual differentiation of the brain in mammals began with studies of this behavior. So here are some rats that are copulating. This is the female, this is the male. And if I drop a female into a cage with a male rat, a lot of things will happen that I can guarantee you. The male will start investigating her anogenital region and her mouth, be sniffing around quite a lot. And sooner or later, he will come up behind her, grab her flanks with his forepaws, and try thrusting his pelvis against her backside. And 3 days out of 4, nothing happens. The female acts like nothing's going on, nothing's happening at all, and eventually the male gets frustrated and stops. Rape is impossible in rats because there's no way the male can get his penis inside her vagina unless she pushes her tail to one side and lifts her up. 1 day out of 4, we say that she is sexually receptive, or an estrus, because on those days when the male mounts her, she shows this posture known as lordosis. Technically speaking, lordosis just means curving your back such that your belly is being pushed out. It's the opposite of kyphosis, when you bend your back so that your belly is being tucked in, right? But in the literature, a whole suite of behaviors that females have to show in order for copulation to take place is known as lordosis, or the lordosis posture. And questions about that? Yeah.
You might cover it more specifically, but if the ovarian steroid is like causing this to happen, it's also dependent on like scenario. Like if you were to just give the steroid hormone without presence of a male, would they still do that, or is it dependent on like your views?
Well, it's a great question. First, let me catch up to you some. So it varies from different mammalian species, but in those species where females show estrous behavior or sexual receptivity, in those cases, it's always ovarian hormones that are typically gating that behavior. And the preview is they show this behavior at the time that's optimum for getting pregnant. Right? So the behavior is being coordinated with ovulation when the ovary releases an egg, and therefore it could possibly be fertilized and can result in pregnancy. Which ovarian hormones, estrogen or progesterone, or both, varies across species, right? And that can vary. In rats, there has to be some estrogen for a few days and then progesterone. So if I want a female rat to show estrous behavior, I can take out her ovaries and they'll be irrelevant now. And I can give her an injection of estrogen on Monday and a second injection of estrogen on Tuesday. And then on Wednesday, I'll give her an injection of progesterone. And about 6 hours later, she'll show lordosis behavior in response to a male, right? And she'll show it for about 5 to 6 hours, and then she won't show it anymore. This is an example of what we say when hormones have an activational effect. Acting in adulthood, they cause the animal to show a reliable change in behavior for a short amount of time, temporarily. So you're asking, will she show the lordosis response if she's in the cage by herself? No. And now I have to confess that there have been times in my life when I needed to see if a female rat would be an estrus. And if I took my 2 front fingers and put them on her flanks and then pushed my thumb against her backside, if she'd gotten the hormone treatment and responded to it, she would lift up her rump for me, right? I know. So I'm not going to put this in writing because I might get arrested. But and then that way I would know that if I put a male in with her, she would show the behavior. If, for example, I was trying to study the male's behavior. So an interesting thing about that case is she's not at all happy with me, right? So she's showing this response despite the fact that my putting my hand in there is stressful for her. So it's, for this reason, in rats at least, this is a lordosis reflex. If I provide the right stimulation, her spinal cord and her body's going to take up posture however she's feeling. If she's stressed enough, of course, then the stress hormone response will overwhelm that. But it's pretty prominent. Okay. Yeah. I know. How many people know there's been hours watching rats copulate? See, there I am. Not lately. So, on the other hand, males, if they are castrated, will stop showing their behavior. So, in females, I can make her show this behavior by giving her the right ovarian hormones. In males, if I castrate the male, the next day he'll still copulate like normal, and for a few weeks afterwards, but eventually after castration, his copulatory behavior will go down and down until after a month or so, he'll stop copulating altogether. He won't show any interest in the female and mounting her. So this is a gradual effect. And so if after that's happened, I start treating with testosterone, pretty soon the mounting behavior will come back. And so this is another example of an activational effect. The testosterone, or other androgens work too, is activating mounting behavior. And in this case, in a gradual fashion, and only for a while, right? If I take away the testosterone, then the behavior goes away. This is a graph I like from a classic paper back in the late 30s, I think. They happened to be looking at guinea pigs, where they wondered whether, testosterone had only just been discovered. And so they're wondering, maybe the fact that different males have different propensity to copulate, maybe that's because of how much testosterone they have, right? And nobody could measure testosterone yet, but they had isolated it. And so they could use it to treat animals. And so they did what other people had done before, and what people who work with animals had known for thousands of years, that if you castrate males, eventually, in various species, eventually they stop copulating. So they castrated the animals, saw that they divided the animals into those that liked to copulate a lot and those that liked to copulate some, and those who were pretty poor, pretty uninterested in copulation. They'd do it, but no enthusiasm, right? And never mind what the scale was. So when they castrate them, everybody goes down to 0, and now they started treating them, and everyone got the same dose. And even a low dose of testosterone got the animals to start copulating again, but notice that they divvied up into their different groups just as they were before. So they concluded that individual differences in male copulatory vigor, if you like, is not due to differences in testosterone levels. Rather, it's due to differences in how they respond to the testosterone they have. And it's interesting that they then tried a higher dose to see if they can make them all respond. And in fact, notice that they still stayed in their different groups, but notice, if anything, copulatory behavior went down. So there's too much of a good thing, right? Too much testosterone is probably interfering with it. So what we conclude from this is that testosterone is important, androgens are important for copulatory behavior in these male animals, but it has a permissive effect. So other people would show that even if you provide just enough testosterone, 1/10 the level that animals normally have, that would be enough to fully reinstate the copulatory behavior. So it's a permissive business. There has to be some testosterone around for the animals to copulate, but more testosterone doesn't get you more copulation. How's that? By the way, if you're wondering, the same is true in humans. So it sometimes happens that humans lose their testes, either for surgical accidents or other accidents, etc. And people have done double-blind, placebo-controlled studies where men who had no testes were given testosterone or placebo. And indeed, the men that were getting testosterone saw a prominent increase in libido, and the men that were getting placebo did not. And it's a little bit, I should also tell you that the men who were getting the testosterone, apparently they all knew, because they felt so much better when they had some testosterone around, right? But again, even a low dose is effective at restoring libido in those men. And it sometimes happens, there's also pretty good evidence that testosterone is important for controlling libido in women as well. I told you females make testosterone too. It sometimes happens that women after menopause complain about having little interest in having sex, and treating them, giving them estrogens that causes many beneficial things for them, but doesn't affect libido. But in fact, instead, low doses of testosterone are the only known effective way of sometimes boosting libido in postmenopausal women. Much lower levels than given to men. Sam?
Started that it seems to be fast testosterone in the future.
Do I happen to know the differences?
And different know what's causing the differences in response?
No, and so even though this was known like back in the 1939, I think, maybe 1943, something like that, we still don't know why males differ in how sensitive they are to this effect of testosterone. People have looked for various things. There's a difference in the concentration of testosterone receptors, actually androgen receptors. No. So, yeah.
If it's testosterone have a gating effect, how come it's not like a really steep drop off? How come it's more?
Yeah, why is it more gradual? The assumption is, I mean, remember, testosterone, it's affecting gene expression, and in general, hormone effects tend to be more gradual, right? If you want something to happen absolutely fast, like within a few milliseconds, you need to have the nervous system involved in that. For these sorts of physiological changes preparing for reproduction, they tend to be more gradual. And so presumably it's the loss of testosterone. When castration happens, because testosterone, the half-life is very short, all the testosterone is gone within about 4 hours.
Okay.
Because there's just no more there. And yet the effects are very gradual. So presumably whatever physiological processes they're having on the brain, among other places, is probably takes time to undo itself. How's that? So we have in mammals, we have a good understanding of sexual differentiation. And as far as I know, across almost all the mammals, the rules are very much the same. So females, as you know, are XX bearing. They have 2 X chromosomes. So the female can only make one kind of gamete. It's got an X chromosome or nothing. The male, on the other hand, has the XY complement, and the Y chromosome, if he makes a gamete with using the Y chromosome, then the offspring will be male. And we know what it is about the Y chromosome that is important for that. It's a gene on the Y chromosome known as the sex-determining region of the Y, or SRY, and it's only present in the Y chromosome. And what happens, by the way, the Y chromosome is much tinier than the X chromosome. There's only about 100 genes on it total. Almost all of them are involved in male fertility, those that are left. And the SRY is normally found only on the Y chromosome. So when that happens, the indifferent gonad will start developing into a testes, and in the absence of SRY, it will develop into an ovary. And then it's going to be that genetic sex, XX or XY, that determines the sex of the gonads. And then the gonads are going to determine the sex of the rest of the body based on what hormones they produce. So mammalian sexual differentiation is that genetic sex directs gonadal sex, and gonadal sex uses hormones to direct phenotypic sex. And here's a cartoon of this. So at 6 weeks of gestation, the fetus, if you look at just the structure, you can't tell whether it's going to become either a male or a female, because the gonads there look kind of like both and kind of like neither. And at that point, it's called the indifferent gonad. It's not that it doesn't care, it's just that you can't tell what it's going to be. And the indifferent gonad will have attached to it 2 different duct systems that will be connected to the gonad at one end and will connect to the exterior of the body between the rear legs at the other. These ducts are named after the anatomists who first discovered them or first described them. One are known as the Wolffian ducts, and others are known as the Mullerian ducts. And so they are providing a gateway from the gonads to the outside world. By about 8 weeks of gestation, though, still very early, right? And the whole fetus is tiny still at this point. By that point, at 8 weeks of gestation, if a Y chromosome is present with SRY, then the indifferent gonad starts developing into a recognizable testis, primarily because of expansion of the medulla or internal portion of the gonad. And in the absence of a Y chromosome, the indifferent gonad will start developing as an ovary, primarily due to growth of the cortical or exterior portion of the indifferent gonad. And at the same time, the duct systems will be changing so that the Wolffian ducts will be maintained and prominent in males, and they're going to provide all the structures that get test sperm to the outside world. And in females, and in males, the Mullerian ducts will fade away. So again, in terms of mnemonics, biology made it easy for you here. You've got to associate Wolffian ducks with wolf-like male behavior, right? Wolf whistles and things like that. And Müllerian ducks start with the same sound as mother, right? So the Müllerian ducks are going to be important for females and the Wolffian ducks in males. In females, in the absence of an SRY, the ovaries develop, and now the Müllerian ducts are going to be prominent and the Wolffian ducts are going to fade away. This process of the gonads directing the differentiation of the ducts is due to a hormone. It turns out that testes make a hormone known as anti-Müllerian hormone. And the name tells you what it's going to do. The testes are going that you secrete anti-Müllerian hormone, which mostly acts locally to cause the Müllerian ducts to go away. And the other hormone that's involved, also coming from the testes, are androgens like testosterone that are going to boost development of the Wolffian duct. The female ovary in these early stages doesn't seem to be secreting much hormone at all. But as long as it's not secreting anti-Müllerian hormones, the Müllerian ducts are programmed to develop. And as long as the ovary isn't producing testosterone, the Wolffian ducts are going to wither away without that steroid signal. And so what will happen eventually is under the influence of these hormones in males, the Wolffian ducts will form the epididymis, which is a structure, the sort of crescent moon-shaped structure right next to the testis where sperm is stored, the vas deferens, and the seminal vesicles. And that's going to provide a pathway for the sperm to eventually get in the outside world so this individual can reproduce. In females, in the absence of AMH and in the absence of testosterone, the Müllerian ducts will develop into the oviducts, the uterus, and the internal portion of the vagina, and that's going to provide a pathway for eggs to get to the outside world. So far, so good. And then finally in this picture, I want to show you that the testosterone from testes are also acting on the outside of the body. And so in the presence of androgens like testosterone, the exterior, which originally had a little genital tubercle at early stages, testosterone will cause that tubercle to grow more in males than in females, and so they'll form a penis from the same tissue that in females forms clitoris. And the testosterone will also act on the epidermis around the genital area to cause the swelling and formation of the scrotum, which will hold the testes eventually in males, and the formation of the labia that surround the vagina in females. And it'll also be the skin here that will form the external portion of the vaginas in females. I mean, it should be more exciting since it involves genitals, right? And yet, yeah. Questions about that? Well, perhaps so again, genetic sex determines gonadal sex, and it's gonadal sex that determines phenotypic sex. And for the outside of the body, at least, the fact that androgen and testosterone and other androgens are important for masculinizing the body is made especially clear in cases when the androgen receptor does not work. So if a fetus has a defective gene for the androgen receptor, and it happens to get a Y chromosome from dad, the SRY gene on the Y chromosome will cause the indifferent gonad to develop as testes. The testes will produce anti-Müllerian hormone, which will suppress the Müllerian glands. And the testes will also make testosterone. But if the body that can't doesn't have a functional receptor to do the testosterone, then it ignores it. And so the rest of the body will develop in a feminine fashion. So the exterior will look like a female, and the interior will look like a male, right? Or let's see, the interior won't look like either male or female, because on the one hand, the no Mullerian duct structures, no oviduct, no internal, no uterus, no internal vagina, but neither are there Wolffian duct structures, because they, even though testosterone is around, they can't respond to that because they don't have an effective receptor. And in cases like this, that sometimes happen, if it's complete androgen insensitivity, the individual is born at birth. The doctor does a incredibly sensitive analysis, looking between the legs, and says, congratulations, you have a girl. The girl grows up just like every other girl, and eventually all of her peers are having their first period, but this girl isn't. She'll eventually go see a doctor who, if it goes see an OBGYN, the first thing they'll do is a pelvic exam, where they'll notice it's a very shallow vagina, and they see no evidence of a cervix. If they take blood levels, they'll assay and see that the individual has higher levels of testosterone than most males. Why? Because they're not getting the negative feedback without the androgen receptor working. They're not getting the negative feedback to turn off the call for more testosterone. And if they do a karyotype, they'll see that they have an XY genotype. And yeah, so we'll talk about that condition some more when we get back. So see you next week.